Hello, my name is Joshua Mugabi. I welcome you to Viruses. Are viruses cellular beings? Viruses are considered living beings, but they do not have cellular structures. There is some controversy regarding their classification as living beings. Their characteristics of self-reproduction and of having genetic material, however, reinforce that classification. What is the basic structure of a virus? Virus constitute of genetic material, that is DNA or RNA, covered by a protein capsule, also known as capsid. Some viruses, like HIV, have additional extra envelope derived from the plasma membrane of the host cell from which it comes. Are there non-parasitic viruses? All viruses are obligate intracellular parasites. That is, they depend on the host cell to complete their life cycle. A virus does not have its own metabolism. Why is it a strong evolutionary hypothesis that although viruses are structurally simplest beings, they were not the first living beings? The fact that viruses are obligate intracellular parasites makes it very weak the hypothesis that viruses appeared before cellular beings in the evolution of life. What is the genetic material of a virus? How does this material act in viral reproduction? There are DNA viruses that have double-stranded DNA and RNA viruses with single-stranded RNA. Viruses inoculate their DNA or RNA molecules into cells. Then these cells, by means of transcription or reverse transcription and translation, synthesize proteins for the assembling of new viruses. This synthesis is commanded by the viral DNA or RNA molecules. What is the typical reproduction cycle of a DNA virus? A typical virus has proteins on its capsid that binds to the outer membrane of the host cell. In the place where the virus adhere, viral proteins act to break the cell membrane. And then the virus injects its DNA molecule into the host cell. Within the host cell, viral DNA is transcripted, and thus messenger RNA is produced. Viral messenger RNA is then translated and the viral proteins are made. Viral polypeptides made within the host cell are cut by enzyme called protease, and then copies of the viruses are assembled with the newly formed proteins. When the assemblage of the new virus is completed, the cell membrane breaks and the viruses are released outside. Unsolved infected cells can produce hundreds of viruses. What are retroviruses? How do they reproduce? And what is the role of the enzyme reverse transcriptase? Retroviruses are viruses whose genetic material is RNA. HIV and the virus for SARS, severe acute respiratory syndrome, are examples of retrovirus. These viruses inoculate their RNA into the host cell. And within the cell, the viral RNA is reversely transcripted into DNA. DNA made from the viral RNA then commands the synthesis of viral proteins for the assemblage of new viruses and the breaking of the host cell to liberate them outside. The enzyme reverse transcriptase is a catalyst of the reverse transcription of RNA into DNA. The enzyme is part of the virus, and it is also inoculated into the host cell. What is the basic structure of HIV virus? What is the function of glycoproteins in its envelope? HIV is an RNA virus. Its core, there are two strands of RNA and reverse transcriptase molecule. The core is covered by a capsid, that is a layer of proteins. The capsid is then covered by the envelope having glycoprotein and lipids. The glycoprotein of the HIV envelope are located on the outer surface of the virus, and they are responsible for recognition of the cells to be infected. The HIV host cell is a CD4 lymphocyte, and for adhesion of the virus to the cell membrane, that is, the CD4 is a receptor glycoprotein, of the outer membrane of some lymphocytes. What are bacteriophages? Bacteriophages, these are viruses specialized in parasitism of bacteria. They are used in genetic engineering as molecular cloning vehicles to insert recombinant DNA into the bacteria. They were also used by the former Soviet Union to treat bacterial infections. Bacteriophages have polyhedral-like capsid and DNA as a genetic material. The head of the virus is connected to the tail that ends in small fibers that help the virus to attach to the bacterial cells and to inject its genetic material into the host. What is meant when it is said that a virus is in inactive state? Viruses are considered in inactive state are those whose genetic material within the host cells without synthesis of viral proteins and assemblage of new viruses. The life cycle of these viruses can be activated under certain conditions, and they synthesize the viral proteins, begins, and new copies are made. The virus that causes herpes virus, is an example of a virus that stays in an inactive state and sometimes activated. What are the main human diseases caused by viruses? Among diseases caused by viruses are common cold, mumps, variola, rubella, measles, AIDS, the viral hepatitis, human papillomatosis, HPV infection, rabies, dengue fever, yellow fever, poliomyelitis, hemorrhage fever, Ebola virus, SARS, severe acute respiratory syndrome. Viruses also cause many other diseases in animals and plants. SARS is a disease that appeared in 2003 within the epidemic features of the province of Guangdong in East China. What types of agent causes SARS? SARS is caused by a virus from a coronavirus group, RNA virus or retrovirus. SARS can be fatal. What is crystallization of a virus? What is the importance of this process? Crystallization is the process of transformation of the viral components into organized solid particles. Crystallization of biological macromolecules, including viral components, is used in the study structural characteristics, for example, through X-rays, laser beam, etc. Thank you very much for participating in viruses. Please do like, share, and subscribe.